Hey team, I think there's an internet glitch from your side. Can I start the session? Is the introductory session done?
Yes.
Yeah, I'll just start. Okay, just a second. I'm sharing my screen. I hope my Indian English won't be a trouble for you guys. Please let me know whenever you are not getting a point. I'll be happily repeating until you understand. Okay? Yeah. Am I clearly audible? And the screen is clearly visible, everyone?
Yes, clearly audible.
Okay. Yeah. And the screen is visible, right, ma'am?
Yes, visible.
Yes, yes. Thank you so much. Good afternoon, everyone. I am Madona Mathew. I am currently working as assistant professor in the Department of Forensic Science, Usha Martin University, India. So today we'll be discussing on forensic entomotoxicology. So we all will be familiar with the term forensic entomology basically. Forensic entomotoxicology is a subfield again of forensic entomology. So let's start with what forensic entomology is. It's a study of insects and other arthropods in relation to criminal investigations here. Majorly the insects will help in calculating the PMI, which is the postmortem interval, which is basically a major part of forensic entomology. So for being a forensic entomologist, a basic knowledge of the life cycle of insects associated with corpse and the succession is very important. Now let's see what exactly entomotoxicology is. Forensic entomotoxicology is a specialized field that combines forensic entomology and toxicology. It involves the study of insects found on or around the decomposing corpse, and it helps to determine the presence of drugs, toxins, or other poisons that might have contributed for the death, and that will lead to the clues regarding the death and will lead to a successful investigation. So these insects, the fauna, can ingest or come into contact with the drugs or the poisons present in the body, and their analysis can provide valuable information. And the information regarding the death before the life of death can also be provided when we investigate and when we analyze these drugs or toxins or poisons. So basically, all this information can help the forensic investigators to determine the cause and manner of death, as well as it can provide legal evidence for the proceedings. So basically, all the clues we got from the analysis of the entomotoxicological evidence uh, can be through the involvement of various techniques which are very common to us, which are very common for all the other analysis like GCMS, we have gas chromatography, mass spectrometry. So these will help to identify and quantify the amount in which the drug or toxin is present. So basically, this field requires a thorough understanding of insect biology, decomposition process, which are the main requirements of entomology as well, along with a little bit of toxicology because it's all about toxic materials. So now, what are the clues or what are the advantages or what are the information the entomology gives? So, forensic entomological evidence, basically all the phonas associated with the corpse of the corpse which is decomposing, will give an idea about the original place of commitment of crime. Also, the primary crime scene along with the secondary crime scene can also be given. Then, whether the corpse has been disturbed after death, before or after. So it basically gives a clear idea about if the dead body has been changed from from the primary crime scene to the secondary crime scene. Then it can give ideas regarding the time elapsed after the crime, the season of death, basically because we all have. Oh, slides are not moving. Everyone, slides are not moving.
Yeah, it's still on slide one.
Still on slide one. Okay, now moving.
Okay, is it all right now? Can you say it?
Yes, slide
Yes,
two.
yes, yes.
Okay, I think when I'm keeping it on the full screen, it's not visible. Okay, I'll continue like that. I hope it's moving right now, right?
Yeah, sure, sure.
Yeah,
Sure,
yeah,
sure, sure.
yes. So we have discussed about what is the introduction about forensic entomotoxicology. Okay, so the other details that entomology gives are regarding the presence of wound, because mostly the all the faunas, all the insects that arrive to the crime scene, or basically the corpse, will first try to find out whether there are natural openings. We all have 9 natural openings in human body. Other than that, if any wound is present, because basically the main requirement of all the fauna which is coming on the corpse is our flesh, our blood, and other fluids which can help them in their reproduction purposes, है ना? So basically they will try to find out the presence of wounds so that they don't have to so it won't be much time taking procedure for them. So if there's a presence of wound, they can start their succession procedure. Then presence of drugs, which we'll in detail discuss in the whole presentation. Then if there is any season related to that, because there are many insects that are only seen during the summer season, and there are many insects or faunas that are mostly seen during the winter season. Then depending on rainy season and other seasons, it changes. So the clear idea about the location and the season can also be then are also available. Then the insect successional wave is employed when the corpse has been dead for a month, between a month up or to a year or more. So basically, what an insect successional wave is, that if the corpse is dead between a month to a year or more, so this ISW, the insect successional wave, is calculated. But if it's below 1 month, the period is below 1 month after the postmortem, so the maggots development stages is used. Next, you all might have known about what post-mortem interval is. It's basically the time elapsed since death. So usually, though, what is the one main advantage of forensic entomology is that apart from all the traditional methods for calculating the post-mortem interval, forensic entomology has been considered very successful because it is the only method as of now which gives accurate PMI even after 72 hours of death. So that's although we have a lot of limitations for forensic entomology and entomotoxicology, and that can be one of the main reason why this field is not that developing in a high rate as of now. In the future, it can be the thing can be something else too. But the accurate identification, accurate detail of the PMI after 72 hours of death can be is one of the major advantage of forensic entomology, which obviously this information is crucial in criminal investigations. Now let's look into what ADH is. So basically, ADH is accumulated degree hours, which can affect insect development. So this is one of the main method used in forensic entomology to estimate the amount of thermal energy available for the insect development over time. Because we all know that insects are ectothermic and they will need temperature. Some will need high temperature, others will need low, but insects are influenced by temperature. By calculating the ADH, forensic entomologists can estimate the developmental stages of insects found around the body and inside the body and also above the body, which can be used to determine the PMI. Different insect species can have specific temperature requirements, as we discussed, that some might require high temperatures while some will require low temperatures, and ADH helps account for these temperature dependent differences. So, by using the ADH to estimate the accumulated thermal energy, forensic entomologists can accurately determine the time of colonization and the time since death based on insect development. So next, determining times of death using insect. What are the major clues? As we said, if the dead body, if the death has happened after a month, at the time has crossed after a month to a year, the insect successional wave is used, and if the postmortem interval, if the time is between the postmortem interval to a month, then developmental stages of the maggot has been used. Then we discussed about ADH as well, where insect succession, different insect species colonize a body in predictable sequence. By studying the insect present and the developmental stages, forensic entomologists can estimate time since death. Whereas the developmental stages of insect found on or around the body can provide clues about the time of colonization, which can be used to estimate the time of death. ADH, as we discussed, is a method used to estimate the postmortem interval based on the accumulated thermal energy required for insect development. Next, let's see quickly what exactly forensic entomology in a little detail is, along with the stages of decomposition. So, insects interaction associated with various stages of corpse decomposition. Why do these insects come and infest on the corpse? What are their requirements? So we all know once the body is decomposing, obviously it is a pool of nutrient cycle, and insect will require nutrient uh, in the form of blood and other body liquids and flesh as well uh, for their reproduction process, for their food necessities, etc. In nature, the decomposition process is accelerated by action. Various organisms, of which insects are the most dominant group. Different insects are attracted to the nutrient in island formed from the decomposing cadaver. So different insects, different animals, they are attracted to different levels of nutrient or different parts of our body or different components of our body, one which comes out or which starts decaying once the cadaver is decomposing. So, stages of decomposition, let's have a quick check on what are the stages of decomposition. First one is the initial stage. Initial stage is also known as the fresh stage. Fresh stage means, as the name suggests, it is basically a very fresh stage where apart from the thing that the body is dead and it's not living anymore, and the postmortem stage has started, everything else is normal. Like there won't be any strong order of decomposition. The body will look normal as we are sleeping or something. So basically, even humans won't be getting the order also. But the blowflies, which belong to the Califoridae family, and the flesh flies, which belong to the Sarcophagidae family, are the first people they to visit. They belong to the Diptera order. They are the first insects or fauna to arrive, because there are certain carrion cues, there are certain signals of smell which are produced, which are sent by the cadaver, which is spread around the environment, but it won't be accessible or to our nose, to human nose, but for sure the fauna will start getting some strong smell. So within minutes of the death, you can find blowflies and flesh flies on the dead body. It's not basically the normal house fly, the Musca domestica, you all know, that are present in our that we usually see at our home. Even though these blowflies and flesh flies look like no, the house flies, they are different because they you might have noticed light blue colored, the flies with light blue colored bodies, or a little bit of differentiated bodies from the Musca domestica. Okay, so the next stage is the blotting stage, as the name suggests. And the initial stage will last up to 3 days, and the blotting stage will start from the fourth day and it will last to around the 10th day. And as the name suggests, blotting stage is where the body is blotted, because the body is dead and the flies have started to come, and they might have started their reproduction procedure also. Here what happens is, in our stomach, so we all know, once we are dead, once we turn into dead body, the first thing to blot or swell is our stomach. The reason is there is an accumulation of gases due to microbial activity, which begins inside our stomach and it starts to expand all over the body. Because we, as normal human beings, we do fart, we do burp. So these are our natural mechanism of our body to release some amount of excess gas which our body is producing. So once we are dead, there's no burping and farting, right? So what happens is, all the excess accumulated gases, they are still inside our body. So what happens is, these gases start searching a way to escape the body. So we hear what happens is, maggots start spreading digestive enzymes, and they start to tear tissues with their hooks in the mouth and spread bacteria throughout the body. As the rate of decay starts increasing, now the smell starts spreading, and strong smells of ammonia spreads. And now what happens, adult flies along with the beetles, mites, will start to reach the cadaver in the blotting stage. Next is the third stage, which is an active stage. Usually the decay stage is considered as one stage, but let's talk about it as two stages, because there are two different things, two different points happening here. So I always would like to study it as two different stages. So first, let's see what active decay is. Active decay starts once the blotting is done. Okay, so it will stay in around like 10 to 20 days, 10th day to the 20th day. Now what we saw in the blotting stages, now the body is all swollen up, it's all bloated. Now what's in the next day, what is like, there the gases were trying to find out a way to escape the body. Now they have found a way, the stomach has bursted, and all the gases are now outside the body, they came outside the body. Along with, the body turns to be flattened with everything that is in your stomach, the creamy consistency, which is like white red colored flesh, along with strong smell of decay, and other body parts are exposed. Okay, so this is the stage where everything is like liquefied. Out of all the stages of decomposition, this is the stage where everywhere it's liquid around the body, on the body. Okay, so liquefaction of soft tissues have started. Now this is the stage where mostly all the mites, insects, because it's an open nutrient cycle, everything is outside the body. Now maximum beetles and the Histuridae family will come, and they'll eat everything up, because there's food and food everywhere. So the infestation will be very high. Next fourth stage is the advanced decay stage. In this stage, it starts at the 20th day and it will stay till the 50th day. Now what happens is, now there's nothing liquid things, okay? So everything has dried up basically. So now once everything is dried up, everything that is left are the hair, the cartilages, and some other tissues, fibers, etc. Now what happens is, usually these mites won't be interested in infesting the corpse anymore. So usually these cheese flies that we usually see are present here and eats up all the flesh which is slightly remaining and which is moist. Okay, now parasitoid wasps will be also there. They try to they act as parasites. They try to eat whatever is the maggots or the larvae or the dead maggots, dead larvae of other insects available, and along with the predator beetles are available in this infesting the corpse in this stage of decomposition, along with the necrophagous species which arrive to feed on beetles and flies and to consume dead flesh. The last stage of decomposition is dry stage, which we say as like the skeleton stage or the mummified stage, which is over 50 days and plus. And the stage will last because, as we all know, skeletons or the bones or the teeth, they are not going to decompose as such. They will stay under the soil for years and years. So now in this stage, what is like, the bones are exposed, there's no flesh remaining. The highest number of species is noticed in this stage. There are animals like ctenoid moths. They arrive and they try to feed on the hair, because now everything which is left are the dried materials. Whatever the nails are there, the hair is there, fiber, then teeth is there. So ctenoid moths try, along with the scarab beetles and Dermisidae family, which is all beetle family. So these try to eat and infest and try to degrade the bones and the other hard materials available. Now the pH of the soil, which has turned into toxic or which turned acidic in the third stage of decomposition, when everything came out of the body in the active decay stage. So in this dry decay stage, all the pH starts to restore. So it starts becoming normal. Okay, and the soil fauna like the centipedes, millipedes, snails, slugs, cockroaches, isopods, they are seeing, they just come and like check out and go back, and they don't have they are not into so much of consuming flesh and all. So they just arrive at the corpse and they visit, try degrading some material, and they leave the site. Now, what are the factors that affect the corpse decomposition? Although we discussed about these stages of decomposition along with the days which these stages last, but everything can differ on the basis of few factors. So there are two types of factors, two categories of factors, like eccentric factors and eccentric factors. So eccentric factors like age and what is the cause of the death, then amount of fat, then in case of fetuses and newborns, due to the sterile condition of the body, the rate of putrefaction is slower, and once the body is obese or fat body, the blotting stage will skip very fast, so it will accelerate the decomposing procedure. So now eccentric factors or external factors like climatic factors, temperature, rainfall, access to body, clothing, then animal predators, these are the external factors. Just imagine a scenario where a body is in the second stage of decomposition, let's say blotting stage of decomposition, and it's above the ground, and a vulture we all know vulture is a scavenger, right? So the vulture has attacked the body and teared the body. Now what happened is the blotting stage period has slowed down or shortened down, and it might now skip the stage of active decay and then directly enter into advanced decay. So this is how animal predators play a major role as factors affecting. Then let's imagine a scenario where a dead body is 6 feet under the ground. Okay, so the amount or the number of fauna infesting, and as we said, the flies reach the dead body first. But in this case, we all know sky flies won't be able to infest under the ground. So the number of fauna will be limited based on the location and the access also. Again, let's imagine a situation where a dead body is wrapped completely with lots of jackets and clothing. So obviously the it's not possible for every fauna with to tear every cloth and reach the dead body skin in very short time. So it will take time, the timings will differ, then the fauna trying for it will differ. So these are the few conditions in which um, the period which we discussed earlier will shift. Then we all know that the bodies in colder conditions, cold regions, they will decompose very slowly, while those in the uh, hotter with more temperature, they will be the procedure will be accelerated, right? Then there are few case studies. We usually, as I'm from India, I usually had a list of case studies from India. So today I just thought of discussing the case studies which are not basically stuck to India. So we all know the Ted Bundy case, right? So the forensic entomology has been used in many high-profile criminal cases, such as Casey Anthony trial case and Ted Bundy case as well. And now there's one major case in which forensic entomotoxicology played a very important role. In 2012, murderer of Australia businessman Herman Rockefeller. Here, forensic entomotoxicology played a very important role in the analysis of Rockefeller's remains, revealed the presence of drugs that had been used to incapacitate him before his murder. Now, there are several factors that can affect the effectiveness of entomotoxicological analysis in forensic investigation. It's not at all, it won't be right to say that if we have forensic entomological and entomotoxicological evidence, we will get accurate results. Okay, everything has limitations, and again, as I discussed earlier, entomotoxicology and entomology are not so developed branches, and they are struggling branches as well. So postmortem interval, as we discussed, that is the time between death and the discovery of the body, can significantly impact insect colonization and the presence of drugs or toxins in insect specimens. There are different ways in which the toxins will react to the body of the insect as well. So that can be one factor. Now, environmental conditions, temperature, humidity, and other environmental factors can affect activity and the rate of decomposition, which in turn can influence the presence and degradation of drugs or toxins. You might all have heard about cocaine, a common drug. Cocaine will accelerate the the production, the succession procedure, where let's imagine a situation where normal fly will take around 4 days for reaching to reach its adult stage. But if the body has been injected with cocaine, the egg might turn into an adult fly in just one day. So it will accelerate the whole procedure. And on the other hand, arsenic will, degrade or slow down the procedure. These are few two examples I thought of quoting. Then insect species and life stage. Different insect species have varying feeding habits and life cycle, which can affect their exposure to drugs or toxins. There are different lives. We have to be very conscious about like at what stage we are collecting the insect as well. Can be larval stage, pupil, or adult stage as well. It can also impact the detection of substances. Then other conditions like body location and condition, as we discussed about, if the body is located in indoors or outdoors, or if it's buried, and if the condition is covered, exposed, it also can influence the type of insects present and their exposure to drugs or toxins. I have again noted one question regarding the dead body. In indoors, in the forms, I'll be surely discussing about that as well in the session. Then drug properties is one major factor, because the drug properties of the drug and or the toxin or the poison, such as stability, the solubility, affinity for insects, can affect its detection and analysis in insect specimens. Then there are sampling techniques and analysis techniques as well. The methods used to collect, preserve, and analyze insect specimens can also affect the detection and quantification of drugs or toxins, along with the analysis techniques, because there are a number of traditional techniques as well which might work for few insects or specimen and which won't work. So insect evidence is such evidence that's limited, and we don't have time for all the trial and error methods, so it will be again a difficulty. Then interactions with other substances. Interactions between drugs or toxins and other substances present in the body, which is already present in the body, or which is already, let's imagine a situation where the dead body is in water and the water is polluted. So there are a lot of other metallic compositions or other toxins in the water. So there are interactions between the dead body, between the toxin in the dead body, again with the toxin outside the dead body, which is present in the water, and so can impact the detection and interpretation in entomological analysis. Because at the end of the day, what if the insect, okay, which has infested on the dead body, okay, and again it went into the water and had some and fed on some aquatic insect. So now the insect is having the toxin of the human corpse along with the aquatic flesh as well. So it will be confusing because that insect is filled, or insect will have a combination of one or more toxins. Now let's get into, now we were discussing about the advantages or the clues that forensic entomology and entomotoxicology can give. Now let's see what are the limitations, as we know that both the branches are not so well developed as forensic chemistry or toxicology or biology or question documents is. So what are the limitations? So first is the variability in insect behavior. Insects have variable feeding behavior. They have different life cycles. They belong to different habitats, which can lead to variability in the types and amounts of drugs found in the insect specimen. Next are the environmental factors, as we discussed, include temperature, humidity, then presence of other chemicals, then post-mortem redistribution. After the death, after the human body has entered into the post-mortem stage, the drugs and the toxins can redistribute within the body and may be redistributed to insects also. It can happen both ways. It can be in the human body, it can get redistributed in the fauna body as well. So this can complicate the interpretation of entomological results, especially in cases where the post-mortem interval has been prolonged. Now the limited availability of reference data, because again, because the field is not that established, there are still very limited data regarding, and literature regarding forensic entomology only. So the condition of forensic entomotoxicology is slightly badder. Okay, then sample contamination can also be a limitation, because insects collected from the environment may be contaminated with drugs or toxins from sources other than the body, as we discussed regarding the dead body which is found in water body or in drowning cases. Now, what are the career options? So forensic entomotoxicologist and toxicologist are one of the main career options if you're interested in pursuing a career which includes both forensic entomology and toxicology. Here, specialization in toxicology, focusing on detecting the drugs and toxins in insects associated with human remains, have to be specialized. Again, research scientists who conduct studies to advance the understanding of insect biology, the decomposition procedures, and the detection of drugs or toxins in insects. Then crime scene investigator who will investigate drug related cases or poisoning cases or toxin cases. These investigators may work with forensic entomologists and toxicologists to collect relevant insect specimens for analysis, because even though we specialize in forensic science, basically it's not necessary and it's not possible for us to specialize in every evidence separately. So if you have an interest in entomology or entomotoxicology, it will be great if you can solely pursue the career in that direction and not in the broad field. These are very limited options. There are further options as well. So now let's get into the question. These are the few selected questions I thought would be good for discussion, which I got from your feedback form. So, in a closed room with no insects presence, if a dead body is there, for sure, it also comes under the factors affecting, because it's an indoor cadaver. Okay, so obviously the amount of the fauna will be limited. Okay, but even if I say it's limited, it's not at all possible. Not at all possible that the cadaver will be left forever without the infestation of any, any fauna. Let's conclude that in some cases, flies such as blowflies or fleshlights may be the first to colonize a body, even in closed environments, because the rooms can still have small openings. Okay, let's imagine a far situation where there are no small openings as well, but still we got anaerobic bacteria, we got microorganisms which will develop, and those will be the fauna to carry out the decomposition process. So basically the whole cycle or the whole set of fauna we discussed during the different stages will be differing, but it's not at all possible that no insect or no fauna will be coming or infesting. There will be still the presence of microorganisms. Again, molecular clock genes, which evolve at a relatively constant rate, can be used to estimate the PMI, the postmortem interval, and the time since death based on genetic changes in the insect specimen. This technique can complement the normal traditional methods of estimating the time since death. Yes, so molecular clock genes can be helpful, and it can be the modern estimation method for the PMI. Now, forensic entomologists can overcome the effect of ammonia compounds on blowfly oviposition by using different techniques to attract and collect blowflies. There are different techniques like bait traps are there, sticky traps are there, and you can actually take those using forceps, or even hand with gloves can be used. So additionally, studying the behavior of blowflies in response to different stimulus can help understand their oviposition preferences in the complex environment, because there are insects with different opinions or different lightliness. So there are insects which are attracted to light, and there are insects which are attracted to darkness. So the collection method of them can be differed as in relation to which and what they are attracted to. Next, okay, now this was one good question I got uh, in the form, which is like, we all have heard about what entomology is, what entomotoxicology is, but what can we do with this evidence, how to collect these evidence. So it's very simple. Proper traditional methods can be used for collecting and preserving, and what to do next with the entomological evidence. So use proper collection methods such as aspirators or forceps to collect insects from the body and the surrounding environment if the insects are dead. Okay, if the insects are not dead, if those are flying, then you have to go with traditional baiting methods or trapping methods to collect those insects. Next, to preserve these entomological evidence, the easiest and the most commonly available preservative can be ethanol, okay, ethyl alcohol, so as to prevent degradation and preserve morphological features for identification. Because ultimately, after catching the specimen, our aim is to identify the whole species, then whole taxonomical levels. So it's necessary to preserve the morphological condition of the insect. Next, identify collected species, insects to species or genus level using taxonomic keys or expert assistance. This is the place where a person from pure zoology or biology or entomology can only help. That is what I have stated earlier, that if you're interested in pursuing this as a career, try to pursue the this as a specialization and not just on a broad level, as we are doing masters in forensic, and you can try specializing in forensic entomology or entomotoxicology, so as to get clear idea about the insects and the species. Then record and monitor temperature changes over to calculate the ADH for insect development. Then document the stages of decomposition, document the developmental stages of insects like eggs, larvae, pupae found around the body or on the body. Then refer to the developmental data for relevant insect species to estimate the time since colonization. Then again, once you do all the above said things, then use statistical methods to analyze the data and estimate the time since death, taking into consideration the insect evidences and the environmental conditions. You should always have a record of the temperature, the humidity, and the climatic specifications of the location. Then, what is the admissibility of forensic entomology evidence? So, we all have discussed about the evidence, and now let's see how admissible it is. Will the court accept this as a entomological, accept this evidence as the main evidence if something else is not at all available, and the only thing that the investigator can provide is an fauna or the details you got from the fauna? So how admissible it is? So So if I'm talking in regards to India, and I can see there are not so much participants from India, but still uh, talking with Indian scenario, the entomological evidence are clearly admissible in court if we consider few factors. And again, not clearly talking only about India. Let's talk about common thing that yes, the forensic entomological evidence are admissible, but certain factors or criterias or standards have to be taken care. The relevance, the reliability, like the evidence must be relevant to the case. And it's not that you are just providing any fauna as an evidence, but the court should get a clear picture that yes, the fauna evidence you are providing is of approximate or accurate relevancy to the case. Okay, then there's reliability. The methods and the techniques you have used to analyze the entomological evidence should be scientifically valid and reliable, and there should be consistency in the results as well. Then peer review and publication, there should be evidence stating that this similar kind of evidence have been used in some other cases, and there should be valid publications in scientific journals as well. Then the error rate associated with the methods or the techniques used to analyze the forensic entomological evidence should be known and considered acceptable by the scientific community. Then the standardization of the tools or the techniques or the instruments which we use to for the analyzing the evidence should be also done. And then qualification of the expert who is giving the opinion regarding the forensic entomological evidence should be clearly having the eligibility with the experience, with the qualifications to give uh, clear ideas and descriptions related to the forensic entomological evidence. So the qualification of the expert is also in count. Then the evidence should be fit for the purpose. The evidence must be relevant to the issues in dispute in the case and must assist the judge in understanding the evidence or determining the fact in issue. Next, to identify the toxins present in the body, we have several methods, collecting methods. The CSI officers can analyze the body fluids. It can be any body fluid, any biological fluid like blood or sweat or saliva, semen, vaginal fluid, anything can be that. While insect may not retain the toxin, it may still be present in the body fluid or tissues of the deceased. Analyzing these samples can help detect the presence of toxin. Then, chemical analysis of decomposed tissues, the tissues which are already decomposed in the or are in the advanced stages of decomposition, which is the fourth stage of decomposition. Still certain tissues have the ability to retain the toxic materials or even the metabolites. Chemical analysis can give results and identify the toxin and give the quantity in which the toxin is present. And several instruments, as we discussed, LCMS, GCMS, these can help in quantification of the toxins. Then toxicological screening, again, toxicological screening of various body samples like liver, kidney, brain tissues using techniques like GCMS, LCMS can help identify a wide range of toxins and metabolites. Then historical information, the information regarding the disease, the medical history, any pills or any medicines the person have been taking, or any drug history before death and after death, or circumstances, the incidences surrounding the death can also provide clues regarding the potential toxins that may have been present. Now let's come into conclusion. Forensic entomotoxicology is valuable field that combines aspects of forensic entomology and toxicology to investigate the presence of drugs, toxins, or poisons in insects associated with decomposing human remains. By studying insect colonization patterns and analyzing insect specimens, forensic entomotoxicologists can provide valuable information regarding the time since death, the cause, and other aspects as well. However, the field of forensic entomotoxicology has its limitations, including the variability in insect behavior, the influence in environmental factors, and the potential of sample contamination, which is main. Because if I'm talking about Indian scenario, I'm not that clear about the African scenario. But what in India happens is, even though we still are in the urge to control the pollution, we still have a lot of polluted areas, and pollution in the sense of, if it's water pollution and air pollution, these actually contribute a lot to the deposition of toxins around nature and on nature. So the potential of sample contamination is very high in this Indian scenario. So thank you so much. This is here I am dropping my email ID in case anybody has any doubts. We will still have a question answer session. You can drop your questions in the question answer in the chat box, or I'm open to answer that. If you still have questions, you can please mail me on my email ID, and I'll be very happy to answer. If I'm still in lack of knowledge, I'll for sure study on that and clarify your doubts as well. I hope the session was uh, informative and my Indian English won't have affected our communication. Thank you so much. Handing over the session to the organizers.
Hello. Yeah, your session was definitely informative. Thank you very much for your time and then the lecture. So now we're moving to our live Q&A session. So before I open the floor to the audience to ask any queries that they have or any question that they have, I would like to start with a question that I have here. You mentioned about the insect growth, like that is the rate of decomposition insect growth being affected by cocaine and arsenic. Where you said in case it is cocaine, there will be like a faster growth, like a higher developmental growth for the insects. And when you have less situation of arsenic, their rate of growth might be decreased. So what influences, like what are the mechanism behind? How does the chemical or the drug affect the insect growth? Can you just take us a little through that?
Yes. So usually when we study entomotoxicology, this cocaine's example and arsenic's example are the main ones we are focused on. Okay, usually the cocaine has various effects on insects. Primarily, it depends upon the concentration. The main thing is the concentration of the drug and the method of exposure. Then there are some research also which suggests that cocaine can be also toxic to insects as well, leading to the increased growth rates and their altered behavior. Again, it's exceptional cases are there can be some species in which cocaine can degrade the growth as well, but the literature study shows that in most insect species, cocaine has been accelerating and arsenic has been decreasing or decelerating the growth procedures, and there are altered behavior, and in some cases of death, and in some cases death as well. However, so this exact impact on the drugs and the exact impact on various drugs on various insect will vary.
Oh, okay, all right. So I
Yeah.
was also wondering, like more often than not, like from literature, when a body is of
Yeah.
higher temperature, like when the dead corpse has like higher temperature, when you expose it to higher temperature, or anything that would expose the decomposing body to higher temperature, will
Yeah.
will indirectly also affect the insect growth. So I was thinking maybe these two drugs have a way of maybe either increasing or decreasing the decomposing body temperature.
Right. Even the normal medicines we take, no, let's take an example of paracetamol. Okay, which is we use for the a fever and all. The normal basic medicine or any painkillers we are taking, let's take Combiflam or Dolo 650, everything will have even either minute changes. All the medicines you have been taking your entire life and whose trace evidence is still present in your body will ultimately have an effect on the fauna which is infesting your dead body. So it can change, the factors affecting will change.
Please, the floor is open. If you have any query, you can just raise up your hands or you put it in the chat box.
Yeah, I think we have
Miss-
we have a question in the chat box. Ma'am, can we say that entomotoxicology is a sub branch of entomology? Yes, as I said in my introductory part, entomotoxicology is a sub branch of entomology because it's all about entomology. It's all about insects, and it's just that an inclusion of the drug which is present inside the insect. So yes, it is a sub branch. Then in case if body is shifted to another place, how will we identify it came from another place? See, we have two crime scenes. Let's imagine two crime scenes: primary crime scene and secondary crime scene. Okay, so let's say primary crime scene was a south is somewhere in the south of a country, and secondary crime scene was somewhere in the north of a country. So we all know that the fauna, the flora, the whole diversity, if in context with India, if you're saying even the language changes. So at the time of death, the first stage has started. Okay, so the insect, the flies which is infesting the dead body in the primary stage of decomposition or the fresh stage of decomposition will stay. Their eggs must be laid in the dead body, and now if the dead body has been changed to the secondary crime scene, that is the second stage, area. So what happens is when the crime scene officer or the investigator collects the evidence, he will for sure get evidence of the egg or the larval or the pupal case, or if in case if the fly has now become adult fly and moved also, there will be some trace which will help the investigator, or some cases or any leftovers by the fauna which will help the entomologist to identify that yes, this insect is not from this area. So the insect, basically the fauna does not belong to the secondary crime scene. So yeah, the availability of different flora and fauna at different levels will contribute to the will help in identifying the location. I hope, Deepti, this answer was clear. Am I audible?
Yeah, very audible. Please, any
Yeah.
more or further questions? Okay, so I think there are no questions, but I'll just give this scenario, then we see how we address it. So let's say you go to the scene and then you find a decomposing body.
Yeah.
You find the decomposing body with all the insect evidence there, some, how will I call it, maggots, the larvae, the pupa,
Yeah.
and some blowflies around it. How would you just do the, and
Right.
you want to run an entomotoxicology, you want to do a toxicological analysis from the insect evidence? Can you just take us through a brief processes from the scene to the lab? How?
Okay. See. Yeah. Yeah. If I'm not wrong, you want to know if you're getting an entomological or entomological evidence, how can
Yeah,
we get clues from that, and how are we take the whole procedure of getting the entomotoxicological evidence, right?
yeah, exactly.
Yeah. Yeah, I hope the slide was clear when the procedure of crime scene management is pretty similar to that we commonly proceed with. Okay, so and once we have an entomological evidence, what we do is the collection procedure, entomological evidence, the collection procedure might be not that different. Just the difference is that how we are preserving the evidence, because the main challenge in collecting and preserving the entomological evidence is that, see, once for example we have collected a egg. Okay, so now it's our responsibility to make sure that either the egg is not clearly developing, okay, because the evidence will be in that stage, or else if it's developing, you have to keep a clear record of the way in which it's going, and you have to still be able to, you have to be able to find what and in what level the drug is present in the body. Then starting with the isolation of the scene, and the normal procedure, you have to ensure the area is being, cordoned off, or again we told about contamination. Right now your main thing is that you have to prevent the crime scene and the entomological evidence from contamination, and again make sure that there's no disturbances happening and unauthorized entries and exits are not happening. Then documentation, the chain of custody should be taken care. There should be proper photographs, sketches, notes. Then location, again, environmental conditions of, relevant environmental conditions like the temperature and the humidity. These two has to be always taken care of. The screenshots or the proper detail of the temperature and humidity at the time you collect the information or the evidence have to be noted. Okay. Then while collecting the entomological evidence, one should be clear about the insect specimen, the species, the clothing, the surrounding area. So again, appropriate tool have to be used, because in case if it's a pupil case, okay, it's very delicate. So it won't be advisable to properly use a forceps, because forceps is a little hard, so pupil case can be damaged. Either you use a forceps with a rubber end, or if you can hold two cotton buds and take slowly, you have to take. So it depends upon what entomological evidence you have, so the tool will differ, the method of preservation will differ. We have only talked about ethanol is common, ethyl alcohol is common way of preserving. You can go for 4% formalin solution also, it is also commonly used as a preservative, right? Then it should be made sure that if the specimen is live, if the insect is live, then the container should be breathable. You should not allow the specimen to die. So the packaging should be a breathable environment. There can be, paper envelopes are there, available pill vials are there. So it will actually allow the passage of, oxygen, and will prevent from the mold growth as well. Then again, if it's dead, if the sample is dead, then make sure you store them in a cool and dry environment. Again, chain of custody have to be maintained, because it's again important for the, procedures and admissibility in court. Then the normal procedures like analysis, we can use GCMS and LCMS as we discussed, and other techniques. Sorry. Then go with the report writing and code presentation. I hope I was clear.
Oh, okay. I think I get it. So basically, like depending on the type of maybe let's say drug that you want to find would how do I call it, would guide the type of, let's say, preservatives, or will tell the process that you're going to use in preserving your evidence, right? Depending on the type of drug and the type of preservative that you would use.
Exactly. The stage in which you are collecting the entomological evidence is the main thing. The rest will follow.
The rest of okay. All right.
Yeah.
Please, any more questions from the audience? You can just raise up your hands, type it in the chat box, or you unmute yourself and talk. Please, if you don't ask questions, the speaker will ask you questions. So ask all your queries if you have.
Okay, please let me
So
know if you have any more questions, because all the questions I saw in the form, which I thought should be answered, I had actually discussed in the question answers slides. So, again, if you have any other question, please feel free to email me. And if you again want my personal email ID, the organizers can give. Yeah.
okay, all right. So I think we would end the session here. But Miss Madonna, any hello? If you are there, just give some few last remarks to the audience in case there are young and upcoming forensic entomotoxicologists or toxicologists or entomologists among us, or let's say forensic science graduates or
Yeah.
students among us. Just some word or two for them, like any remarks from your experience.
Yes. So here I can see my students are also here. I actually teach in Usha Martin University, Jharkhand. I've seen my students have also joined. I will again repeat the words I always say. There's no branch in forensic science as such. It's that the chances or the openings are too high, and there are no underdeveloped uh, branches as well. If it all depends upon your likeliness, because I have been liking forensic entomology since last 4 years. And again, yeah, I'll repeat the point that yes, we still are under the need of the data, and the quantitative and qualitative data is less. But if you're interested, you can still build the data of your own, and in maybe in next 10 years nobody will be saying that the data is less or the literature is less, because you have made it for them. So it's like if you are in properly into any branch, not only in forensic science, any branch, make sure you build your love for that branch. And if you're clearly interested, pursue that as your specialization, regardless of what it has to it will offer, or it's offering. The future can be different. And yes, forensics is a very growing field, and we for the next 10 to 15 years, we don't know which branch will be on top and which will be on the bottom. So basically, if you are interested, go ahead with your branch. Try pursuing different specializations. Until if you are confused, try learning about different topics as well. And again, you'll get lots of people to help you. There are lots of established speakers or established researchers worldwide. So there'll be somebody to help you at any point. Yeah. Thank you so much. Thank you so much, the organizers, the forensic post, for having me, because this is basically my first ever international lecture I've given. So the forensic post lecture will be always special for me in my entire career as well. And once it goes into my CV again, your name will forever stay special. Thank you so much.
All right, it's a pleasure having you. So before closing, I also like to send a huge appreciation and thank you to Miss Madona Mathew for honoring this event and then attending and also sharing a lot of information to us on forensic entomotoxicology. Myself, I've never paid attention to the field until you sent us the topic, and I was very, very impressed by the abstract that you sent to us. And then throughout this lecture, I've learned a lot of things. I believe our audience have also learned a lot through this information and through this session, and have taken a lot of takeaways. As she just rightly said, her email is that you can always reach out to her anytime, any day, and she would assist you. She will respond to you promptly. So, Miss Madonna Mathew, thank you so much for this wonderful session. And to our wonderful audience too, thank you so much for staying with us from the very beginning till the end. Without you, this session wouldn't have been able to come on. We appreciate your time, and then we hope you learn and then you keep on staying with us. we keep on staying with us as we have a lot for you. And I would like to just make this announcement. Our next session will be coming on the 23rd of March, that is next month. So just stay tuned, follow us on all our social media handles to stay updated. We'll be keeping you informed if you follow